Before today's advanced next-gen technologies, there was one revolutionary method that changed genetics forever: the Sanger sequencing. Developed by Frederick Sanger in 1977, this technique became the gold standard for reading DNA, unlocking the code of life, and even making the Human Genome Project possible. In this video, we will explore how Sanger sequencing works, why it is called the chain-termination method, and why it's still used in labs today despite newer methods. Let's decode DNA one base at a time. This technique relies on some principle. For example, this technique is based on selective incorporation of chain terminating dideoxynucleotides or DDNTPs during the synthesis of DNA. DDNTPs lack a 3' OH group that prevents the formation of the next phosphodiester bond, thus stopping the DNA chain elongation. By producing DNA fragments of various lengths and detecting the terminal base of each fragment, the original DNA sequence can be deduced. And this is how the Sanger sequencing works. Let's look at the components that are required for this method. First one is DNA template, single-stranded DNA to be sequenced. Second one is primer, short oligonucleotide complementary to the template's starting region. Then DNA polymerase, the enzyme that synthesizes new DNA strands. Number 4, deoxynucleotides, that is DNTPs, DATP, DTTP, DCTP, DGTP, which helps to form normal elongation. Number 5 is dideoxynucleotides or DDNTPs, DDATP, DDTTP, DDCTP and DDGTP. These are labeled, that means radioactive or fluorescent nucleotides that helps in the detection and cause the chain termination. Along with that, some buffer and magnesium ions are required, which help to optimize the enzyme activity. Here you can see the picture and the difference between the dideoxynucleotide and the deoxynucleotide. Let's talk about the steps that are involved in the Sanger sequencing. The first thing is the reaction setup. 4 separate tubes, each containing DNA template, primer, DNA polymerase and all 4 DNTPs and one type of radio-labeled DDNTPs. Second thing that is required is DNA synthesis and termination. Random incorporation of DDNTPs terminates strands at various positions. Third thing is gel electrophoresis. DNA fragments separated by size on a denaturing polyacrylamide gel. And the fourth one is autoradiography. Gel exposed to X-ray film to visualize band and read the sequences. Here you can see the image of original radioactive method, where in the tube all the things we have to put, and with the help of one DDNTPs, one chain will be terminated, and that's how we can see the sequencing. Let's talk about modern automated fluorescent Sanger sequencing, where single tube reaction is required. All fluorescently labeled DDNTPs added together. Then in the cycling sequencing, PCR like thermal cycling to produce the terminated fragments. In the capillary electrophoresis, fragments separated by size in thin capillaries. Then the laser detection is done, where laser excite fluorescent labels. Emission spectrum identifies the terminal bases. And lastly, data analysis is done. Computer software converts signal into chromatogram or electropherogram and the base sequences. Now, how to read the sequence? In the manual method, we have to read the gel from the bottom, that means the shortest fragment, to the top, that means the longest fragment. In the right hand side, you can see the picture of a gel where the sequence will be reading from the bottom to the top. That means CAGC, GATT, and the sequence follows like this. In the automated method, software assigns bases from the peaks in the chromatogram, as you have seen in the previous picture. The application of DNA sequencing is small scale DNA sequencing is required for plasmid or the PCR products. Then by the help of this method, mutations can be detected. We can sequence verification after cloning. We can use it for clinical diagnostic for genetic disorders and phylogenetic analysis of short gene. This method is very advantageous because it has high accuracy, reliable for targeted sequencing of small DNA regions, and long read lengths compared to most next generation sequencing methods. But with these great advantages, this method has some limitation. It has low throughput and not suitable for large genomics. It is expensive and time consuming. It requires high quality DNA template, and it has read length usually limited to 800 to 1000 base pair. So that's all for this video. I hope you understand. Thank you for watching.